Hello everyone, this is Dr. Maneza. Good morning. It's Thursday morning, September 30th, 2021. And the weather in my hometown is really very good. It feels like it's gonna rain today. So this is the episode 2 of our TechTalk series. And before I begin, I would like to emphasize those of you who are in Dubai or planning to visit in coming 6 months, do visit Terra, the Sustainability Pavilion. Coming back to our topic, light for life-saving technologies. Light is essential for life, we all know it, but since when it became an integral part of medical technologies, and how cutting-edge technology today is utilizing it for a wide area of application, diagnostic, imaging, and therapeutic. Very few of us know it, right? It was Niels Finson who first used UV-based phototherapy to treat lupus vulgaris, and it's a kind of cutaneous tuberculosis. And it was back in 1903, and he also won a Nobel Prize on it. Later in 1960s, laser technology paved its way in medicine, and now laser-based surgical procedures are routinely performed in ophthalmology, urology, gastroenterology, cardiovascular, and dermatology departments. So this huge application of light in medicine definitely makes one think, how God, one light, so many applications, right? Let's first take a mechanistic look on light matter interactions. So electromagnetic spectrum span across a wavelength that starts from hundreds of meters and it narrows down to 0.01 nanometer, like in case of gamma rays. We all know this, right? X-rays, they have the range of 0.01 to 10 nanometer, and gamma and X-rays are used in CT and PET scanners. UV light has range of 10 to thousands of nanometer, while IR span across 0.01 centimeter to thousands of nanometer. We all know the visible spectra, it's somewhere from 400 to 700 nanometer, and radio waves got the largest wavelength, 0.1 centimeter to hundreds of meter. So the reason behind recalling the range of wavelengths of electromagnetic spectrum is light of different wavelengths interact differently with human tissues, and there are only two underlying principles, scattering and absorption, which are used in any technology. So the scatter light is used in diagnosis and imaging, and the light absorbed cause emission of molecule-specific luminescence, or it undergoes inelastic scattering. The emitted light carry information about microstructure and molecular content of the matter, more specifically biomatter it just interacted with. And this phenomena is widely used in optical diagnosis and imaging. So it's the endogenous biomolecules which are sensitive to light and impart therapeutic effect when interacts with light. In medicine, optical parameters like wavelength, exposure time, beam area, energy, power, intensity, and fluence, which is basically energy per beam area, are required to be optimized. For example, in case of laser-induced hypothermia, fluence of 25 will ablate 100 microns of tissue layer at approximately 100 degrees centigrade. Penetration depth is another important parameter. For UV, it's 50 to 100 micrometer down the skin. IR and blue light digs 2000 nanometer, and the deepest penetration we see is in the case of red and NIR, near infrared radiation. It's approximately 1 to 3 millimeter in human skin. Depending on type of light and optical parameters, you induce photothermal, photomechanical, photochemical, or photobiological effects. Our focus today is on laser-based surgical procedure, optical diagnostic, and imaging and we're gonna also explore some emerging technologies based on light. Laser based surgeries are routinely performed to treat cutaneous disorder or to ablate tissues, and it shares global market of $3 billion. Ablation of corneal stroma. It's performed through laser-assisted in situ keratomeleusis. We also call it LASIK, and a neodymium glass laser with a wavelength of 1055 nanometer is used with exposure time around 600 frames per second. And a neodymium doped yttrium aluminum garnet laser is used to treat cataract, which is a leading and preventable cause of blindness worldwide. Pulse dye laser PDL, a 585 nanometer wavelength with exposure time from 0.4 to 2 milliseconds with a fluence of 5 to 10. It's used to remove skin marking in dermatology. So it's kind of a cosmetic application. We also have a carbon dioxide laser to reduce wrinkles, pimples, and scars. It's widely used by cosmetologists. Neodymium doped yttrium aluminum garnet laser with 532 nanometer. It's used to treat benign prostate hyperplasia. It's a condition that affects 50% of men above 50. In urology, laser assisted lithotripsy. It's used to remove stones from urinary tract when ultrasound-based shock waves are not sufficient alone. And we all have heard about photodynamic therapy. It's used to treat many disorders, and it has a market share of $630 million. Ultraviolet B rays. It's been used to treat psoriasis for 9 years, and it's a clinical condition and a skin disorder, autoimmune inflammatory skin disorder that affects tens of millions worldwide. In photodynamic therapy, light interacts with photoactive molecule and induce therapeutic effect. Use of light in diagnostics. Take an example of point of care devices, for example, a pulse oximeter, and what we are measuring is absorption difference of oxygenated and deoxygenated hemoglobin. And if you talk about molecular diagnostics, you see light microscopy. Every day we use it to study a morphology of blood cells. And then there is a fluorescent activated cell sorting technique, which is basically a molecular phenotyping method to diagnose blood and immune disorders. Enzyme-linked immunosorbent assay, which is also a color change technique or a light-based technique where interaction of detecting antibodies and biomarkers result in the change of fluorescence. In surgical imaging, we have this fluorescent guided surgery, which is used to resect high-grade gliomas. So basically, this 5-amino levulinic acid, which is orally administered, and it's metabolically activated from protoporphyrin 9, accumulates specifically in tumor tissues and produce red fluorescence. So it's an easy way to look at tumor tissues. And now we have this near infrared trigger drug release mechanism to treat or to kill a cancerous cell. So what we do is we synthesize nanoparticles from NIR sensitive polymers or liposomes, and they facilitate on-demand release, which reduces metastatic spread. Few emerging techniques. We have this now wearable oximeter, which is flexible, all organic, based on optoelectronics, and it can be worn in skin to monitor blood flow, and precision is close to the conventional ones. Implant market share a big chunk of medical device market, approx $200 billion, and prosthetic retinas, which is based on optoelectronic, are now being used to restore vision. So the future is really very bright. We have this implantable LEDs, and maybe in near future some other devices which are self-powered and harness kinetic energy generated by muscle movement. From photonics, we now have biodegradable waveguides which can be used in light activated therapy, and multifunctional optic fiber implant can be used for neuromodulation in brain. So you see, diagnosis and treatment of many life-threatening diseases is nearly impossible without light. With this, I wrap today's podcast episode. Goodbye, everybody.